wanted to say that today, first of all, I am super interactive person who is on headphones, is not going to be here with me in this vein at all. I teach in a really interactive way. I'm not a TED Talk person. Like, I don't want to speak to a bright light. So please just interrupt me. Otherwise, there's really no point in doing these kind of smaller tutorials. The other thing is that I basically, I don't have a ton of math in this. I'm happy to come back and add, or I can send slides with more. Because for me, I feel like I don't know what people's backgrounds are, but I guess if you're up here, you're probably pretty good at the math part. And I think for me, what's been really important is having intuition about what's the right math, like what's the right way to think about this. So that's what I'm trying to do here. And so if it feels like, why am I sitting through this stuff that's all intuition? That's why it's intentional, and we can shift if we need to. Let me give you a little background. So myself, just so you know where I'm coming from, I was a computer science PhD who wrote proofs, never touched data until I started my first postdoc. That was a long time ago. I have since become very data science-y, and I work a lot in immunology, like Elizabeth, actually. And I don't come from physics. So you will tell as soon as I dip into anything physics-y that you'll see that is not my background. I come from computer science and then biology, and then I got dragged into some physics stuff because it seems really useful. So my group at University of Chicago, we do a lot of different things, but basic idea is complex biological systems, a lot of single cell RNA-seq data, getting into other kinds like spatial data, multimodal data, imaging, et cetera, and lots of development of methods and application of methods. And I'm not one of those people who cares that much if I invent a really cool method. I just want to solve the problem. Sometimes that means inventing a cool method. Sometimes it doesn't. I think that pretty much covers it. This is a recent picture of my group, but kind of recent. We're at a tipping point here. So I had 5 graduate students graduate this past year. So all of my pictures and all of my slides, this is my caveat. I start with a big introduction to this topic by analogy, and all of my pictures are old because they're from before my group disintegrated into, well, no, it didn't disintegrate. It's just very different now, and there's only half as many students. So, right, so a bunch of people are gone. Okay, so that's the introduction to me. Can I just get a feel? You guys are all what? PhD students, postdocs? Who's what? PhD students? Postdocs? The rest of you? People off the street, you're welcome. Okay. And who's physics background? I could tell from looking math background. Great. I was a math person before I was a computer science person, so I'm happy there too. Biology, amazing. It's like this room is like my group. Okay, so that is my group is exactly like this. So one of the things that you can do if you want to kind of try to categorize what is happening in my group is think about whether students do laboratory research. There's the 3 on the left that are in some form of a lab actually collecting data, and there's the rest of us that are analyzing data. And there's some of us who never want to think about biology if we don't have to, and then there's lots of people in between. So that's kind of how this is a snapshot from a year ago. That's a pretty accurate vision of about a year ago. And so one of the things I tend to do, both in immune cell biology and in my general gist of things, is I don't like discrete categories. I tend to put things on a spectrum, especially when I think they really belong there. So that's also true for people in my group, right? There's actually quite a spectra of students in terms of their motivation. And I promise this all has a really good analogy to what we're actually going to talk about in terms of, mRNA analysis. So there's people, like I said, who are really math computing oriented and feel really uncomfortable as soon as they say a biological word, and vice versa. So this kind of comes into, you can think by analogy, right? You can think about if you see a continuous mixture of cell types, right? Cell states, it looks something maybe there might be an axis that looks kind of like this, right? How inflammatory they are, if you're in an immune kind of setting or something like that. So often there's some sort of, or several axes in which you can kind of imagine, cells are sort of placed depending on how active some process is in them, for example. Okay, so right, the question really is, how might we learn something about this structure from the observable data that we have, right? That's also along the lines of what Elizabeth is talking about. And so when I wrote this initially for a very non-math audience, and I was thinking like, what is the equivalent to like transcripts? And I was like, maybe Slack posts or something, you know, like little bits of output that grad students actually generate that sort of indicate what is going on in their brains and what they're doing with their days, sometimes more than they would like. So, right, if I think about, you know, what are the kind of terms that characterize their Slack posts, I might think like cell, gene, tumor, patient, right? This is like the people who are working on data. They're either gathering it for mice or humans in immune settings. And the people on the other side, what more of their words are like data, model, embedding, Wasserstein, along the lines of what Elizabeth said and so on, right? So this gives me sort of, this is if for people who know about, which we'll come to, kind of older sort of natural language processing models, you might think of just like a bag of words type model for each student, right? And everyone's kind of somewhere in the spectrum. And I have this great student, Hannah, who's graduated, but she, I would say, was one of my early like definitive splits. She was very, very passionate about applications in cancer, knew a lot about it, also had math background, knew a lot more math than most people who are passionate about cancer and so on. So like, and those people have kind of a mix of different terms in their posts. And if you knew about all of these, you might sort of infer that there are these kind of two different genres, and then there's like some mixture genres, right? Some mixture people. Okay, great. So this prompts a question, which I think is taken for granted actually a lot of the time in people who work in development, like Elizabeth and many other people, although she's also working on questions that I think are extremely, like they're super interesting to me in the immune system now, because she's looking at response. But in development, you know, there's an assumption that there's a dynamic process that underlies those kind of continuous transitions, right? And that's how we infer it. That's not always the case. So one question might be here, is there a dynamic process that's shifting trainee positions? Like, do trainees actually move along that, or do they just kind of grow up? You know, are they born sitting in some position on this? And what this is really telling us is that, you know, there are people with different features, but there's not actually like a process for moving somebody along this axis, right? This axis. And actually what I've observed, I've only had my group for, I don't know, 6 years or so. But sorry, I just got way warmer. But I've seen that people do sometimes change their research motivations quite substantially sometimes. But you know, if all you saw was, you know, kind of where people are in these profiles, you wouldn't know, right? You wouldn't know in particular whether somebody's moving to the right or to the left, and you know, if they're changing position at all, right? So this analogy also came to me because I was thinking about somebody who came in, Frank. He came from chemistry program, super biophysics-y kind of. And then one day, you know, I started seeing his, hi. Sorry, I'm always late usually. Usually you can count on me to start late.
We couldn't find this building.
Oh, I'm sorry. Yeah, I know it's a pain because the addresses, there's multiple addresses.
Yeah,
anyway.
it's been about us 15 minutes.
That's okay. I'm sorry. I mean, for me, it's okay. I'm only on time because I came here early. That's the only way I'm ever on time. So, right. So the question was, you know, what if we could detect small, nearly instantaneous changes? Could we then sort of tell if students are actually changing position, if they're moving one way or another, right? So I was thinking about Frank because, like I said, he came in, he was from chemistry, he was very physics oriented, and suddenly, you know, he started maybe in his 4th year posting all these papers and thoughts about cancer and immunology. Even his AI papers were really related to cancer immunology. And so if you saw that uptick, right, you would be like, oh, he's maybe shifting a little bit in terms of what motivates him and what he's interested in. And you would maybe start to look for contextual reasons as to why that shift is happening, right? And that's part of what's really interesting about trying to be able to infer directionality. It's not just to get the, you know, the whole axis right. It's about trying to get the actual, you know, trying to learn, maybe potentially infer something about causation. Because if you can sort of check what direction things are moving, then you might see if whenever they start moving in a certain direction, there's some, of course it's, you know, associative still, but you could at least hypothesize some sort of cause that you could then test in a different way. So you might hypothesized that when I put these two, one of whom is an experimental cancer immunologist and the other of whom is super, computational, I put them together working on something, that they had a big influence on each other. And in particular, Frank started moving really much more to the applied side, to the translational side, which I think is the reason, but who knows, maybe it was just, he started a different diet. It's hard to tell these things without lots of testing. So, the point for me is that we can develop testable predictions and hypotheses, and we could potentially try to engineer an environment that creates the change that we want, right? So that starts to sound like medicine. And indeed, that's one of the major applications that I'm interested in. So I want to make a point that sounds funny, but is, again, I promise, relevant, right? Which is that there are lots of continuously sort of variable features. I'm using continuous in the non-mathematical sense, right? That have different dynamics. So for example, there's facial hair differences among all of these people, and that lies on a continuum. But if you were trying to learn the dynamics of that continuum, you would probably get a lot of noise, a lot of sort of weird information if you included the people who never generate any facial hair at all, right? They don't participate in that process at all. So many of these women don't generate any facial hair. And so if you wanted to learn it, you would really want to focus on the right people, the people who have a chance, right, of generating it, and maybe are, shaving it so you don't see it, or maybe they haven't matured yet, so they're still not generating it. Either way, you would know you would learn more from focusing on those people, and that becomes relevant also. Okay, so all of this is relevant to many different kinds of immune contexts and other contexts, but in particular, it's relevant to the tumor microenvironment, which is really when I started thinking a lot more. I'm not a cancer biologist by training, but I have started working much more in cancer. Again, kind of like physics, just got pulled in that direction. And one of the things that I think is really interesting, although there's other really interesting environments like this too, like the gut epithelium is also like this, right? They're very dynamic, constantly changing. And so you don't think about them in the same way you think about development, where there's like a canonical direction that everything moves and they end up in the place unless you really mess up something, right? There's lots of things happening. And in fact, it's kind of hard to study them even experimentally, because sometimes with treatment, you get a kind of similar outcome, but the raw sort of spectra of different outcomes is really complex. Like you get all different kinds of phenotypes in terms of just the cells that are there, what they do, and how they change. And one of the things that we're particularly interested in is the immune component, because we know that can have at least a really large effect on many tumors. And we'd like to be able to engineer that so that you can make it more effective, potentially, right? So that's a big goal of medicine. So one question you might ask is, how can we learn about the dynamics, whether cells are changing, if so, in what direction, and why, right? So this is an underlying question that I'm thinking about. Any questions so far about all of this, you can ask me. I thought another one, like facial hair, by the way, is height. I was thinking if you have full-grown people, right, they have a spectrum, but I wish I could become taller, but I'm not going to. So, yeah. Any questions about this? All pretty intuitive, right? Okay, so one of the things I'll come to at the end, I'm going to try to talk a little bit about it in my talk on Thursday, if some of you are there, is that, so we're going to talk about RNA velocity, which Elizabeth brought up. Many of you, has anybody heard of it before Elizabeth's talk today? And do you have like horrible associations with it?
Yes.
Yeah, exactly. Everybody thinks it's trash because it's usually trash, right? It makes pretty pictures, usually trash. So, I mean, I've sort of found unwillingly ended up working in this area, and I feel like I a little bit drink the Kool-Aid. Like I think there's actually some really interesting signal there if you can kind of learn it properly. I don't think the right place for it is necessarily in learning major transitions, like major longitudinal transitions. I think generally it doesn't seem to do great at that, as Elizabeth pointed out. But it can be really useful when those methods fail. And I'll show a couple of examples. And so I think it's useful as a tool in a much bigger toolbox. And one of the things I'm thinking about as we go forward is kind of thinking about perturbations. And, you can think about perturbations in a kind of natural way, right? Like we think about our genome as kind of being a form of natural perturbation of different genomic context. But you can think about spatial kind of context, like tissue, as also being sort of natural a natural landscape of variation, right? Which cells are near which in different people or at different times in different tissues. And so you could start to try to learn something from that variation if you could actually get, relatively accurate change, I think. Okay, so I'll come back to that a little bit. This is very much work in progress that most people that part is, not something that I should really talk about, but I am bored with talking about stuff that I already did. So, okay, so as this is sort of the non-physics based trajectory inference picture, right? If you think about single cell RNA-seq, the reason we think about being able to do, there's two ways in which you could think about doing trajectory inference. So one is you have clear time point data, like longitudinal data, and you are trying to kind of map, the distribution of cell states at one and see how they progress through time. That's a really great application of optimal transport, for example. It's one of the earlier applications. Another is that you have a distribution even within a single time point, right? So even the hematopoiesis kind of data that she showed, like you actually get a spectrum of all the cells at all the states if you do your experiment, right? And so you could, in theory, capture the full dynamics in your data if you could, infer them, if you knew them. The problem with, or very many early methods did just similarity-based trajectory inference, right? They just took two profiles and said if they're near each other in, sort of similarity by some distance metric, then they're probably near each other developmentally, say, or in transition space. And so you can kind of just link things together like that and you get some kind of chain, just like the dark brown to light brown, right? So is that concept at least familiar to people mostly? Okay, you don't all, it's okay if you're like the one person who is shaking their head, like you can also stop me, I don't care. So the problem with this kind of method is that it breaks down in places where you, first of all, may not know direction, right? So development, we know a lot about development. We know a lot about cell differentiation. So we often have pretty good prior on like where early states are versus later states. And a lot of those methods, basically the major signals tell you the right things. It gets harder when you don't have a lot of information about where things are starting or where they might be going, or if they're going backwards or forwards, or can they change directions? Like the less you know, the harder it is to figure out, for just similarity to be enough to tell you really about a system, right? So RNA velocity is this idea that, the other thing we might not always have is this longitudinal data that Elizabeth said. So for example, if you're taking, tumor biopsies from patients, you're not gonna be able to take more than one from any patient. You're not going to synchronize them all. You're not going to have, right? So you can try to do what you can with mice or other kinds of organoid models, which might help a lot to eliminate that. But there's a lot of cases in which you're just not going to have great longitudinal data. And so you could try to build models like she was saying and try to predict. I think, here what I'm doing is kind of ignoring that a little bit and just asking what can you learn about how things are changing from one sample, say, or from a collection of samples, but trying to infer more instantaneous change. So this velocity model is built exactly on this sort of random observation that, a really smart guy made, which was like, hey, in these data, there's unspliced and spliced transcripts. Often. That's not always true. And in fact, it is less true when it's probe-based, which some of the spatial methods are. If that means nothing to you, it's fine. All you have to do is some have more, some have less of the unspliced. Traditional single cell like 10x has a lot, actually. It's not as well calibrated, and so it can be subject to lots of biases. But the idea is that, you have information about an earlier stage of the process from the same cell, right? So genes have to go through this process where they get transcribed, and then they get, then they get spliced. Exons get, the introns get taken out. They get 5' caps, like all this molecular stuff happens to them, and then they get translated. And we actually have information sort of from the pre-mRNA and the mature mRNA sometimes for at least some of the genes and some of the cells. So that basic idea is like, okay, that's a little bit of dynamic information. If we could use it and we're smart about it, we could infer something. when maybe it's hard. Okay, so we'll talk a little bit for the rest of the time about this RNA velocity, why it's challenging, at least some of the things that we noticed about why it's challenging. It's challenging for lots of reasons, but at least a couple of the things we noticed that I think are more generally interesting principles, how we address some of those challenges. I really liked what Elizabeth did about presenting lots of work, but that was actually not the prompt I was given for this. So I'm sorry, this is a little focused on my own work, but there's a ton of work in this area. And just why I think it might be interesting in the context of spatial, which if that's only if we have time, really, this is just the background. Okay, so basic idea for doing trajectory inference via RNA velocity estimation is that you're first going to estimate gene-specific velocity parameters. And they're pretty simple. The basic model is extremely simple. It's like an ODE, super simple. Transcription, splicing, degradation. You assume, you make lots of simplifying assumptions. And this model has been made a little bit more complicated in various versions to a lot more complicated. It can be, there's a stochastic version of it. But I think it's useful to just think about the most simple model first. So most simple model is just, do I even have it written in here? I got rid of the actual simple model. Sorry. Most simple model is a I'm sure I have it somewhere. I'll grab it for you. Is a very simple ordinary differential equation that writes basically, the amount that you have at any given moment is a function of, your transcription, and then for the amount that if you've spliced, right, that affects how much unspliced you have. That means that you have less of the unspliced. And I will find the very basic equation somewhere. But the basic idea is that from those, you can learn something about, you can learn some kind of phase portrait of where cells are for a given gene, right? And so this is a gene that at very basic level has positive velocity because it's like above this sort of inferred equilibrium between the two extremes of nothing and kind of even amounts. And so a basic idea being that if I'm generating tons of unspliced and my splicing machinery hasn't caught up, right, because I'm just turned on this gene, I'm just upregulating, or I'm trying to generate even more of it than I had, then I would have relatively higher amounts of the unspliced versus the spliced. If I'm turning it off, then it would be inverted, where the splicing machinery would, that I would have stopped transcribing, but my splicing machinery will still need to chug its way through all of the stuff that was already existing. And so I would have relatively higher spliced to unspliced. I would be on the lower side of that. Super simple model, right? Known to break in all kinds of situations. It's actually kind of miraculous that it works at all, frankly. But one of the things that we noticed and others have noticed, including Lear Pachter, who is coming, is that this model, one of the problems with it is that it ignores stochastic transcriptional bursting. So this is known. I'm not like I said, not a cell biologist, not a molecular biologist, really. But basically, we know that cells don't sit there and steadily chug and make transcripts the whole time at a steady pace. They basically make a bunch of transcripts, stop, make a bunch of transcripts, stop. So they have these bursts of transcription. And you can imagine that if your model doesn't account for that, then you might see a bunch of transcripts and think, oh, something got upregulated or downregulated because of this, when actually it was just a burst, right? And it's actually just its steady state doing its bursty thing. So it seemed like this could be a potential source of error at minimum. And we do incorporate a very simple burst model to try to take care of that. Another thing that we realized that really bothered me because of other work that I've done relates to this whole idea about the sort of the student analogy and research motivation. The idea being that just the distinct dynamic processes might be happening in a single time point, right? There's guys growing facial hair. There's, people writing Slack posts about their research, like different processes are happening at the same time in every living organism, including in cells. And basically the way that a lot of methods work right now is they basically dump all the cells that you might have into an inference to try to infer these parameters. So if you have a bunch of cells that are not participating at all in a process, they really dilute the signal and compared to the cells that are. Just like if you had, 3/4 of your group were women and you're trying to learn something about facial hair, it might not be the best, right? You might have very weak signal on facial hair. So that's one thing. The other thing is that, we tend to learn all of the genes also at the same time, learn parameters for all of the genes. And it's clear that, there's going to be processes that are going to be more active in certain cells, and you want to focus on the genes that are important for that process. But of course, this is also obviously a bit of a chicken and egg kind of thing, because, if a process is low, you still want to capture that dynamic, right, of it kind of getting turned on. And so you don't want to exclude that possibility that a cell is just at the beginning or end of some process. Yes.
So earlier you were talking about trajectory.
What's your name? Sorry.
Stanley.
Stanley. Okay, cool.
Nice to meet you.
Nice to meet you too.
So in the trajectory inference framework, you talked about the ordering. So when you're talking about distinct processes.
Yeah, we didn't get there at all.
Yeah, so then the point is maybe that there is a particular ordering within each of these processes.
Absolutely.
Okay.
Absolutely. That's right. And we really don't delve into that. I actually think that would be, so we do a little tiny bit, which I'll get to in a minute, or later on, but yes, absolutely. Different processes have their own view of kind of how the cells, where the cells are, and sort of the up or down regulation of those genes, right? And when we're trying to actually infer a sort of global picture of velocity, we basically kind of cheat, and I'll explain how we do that, how we get some kind of global picture that kind of takes an average over these. But that's definitely, I think that's what's happening, actually. And it's very likely that sometimes they're more or less synchronous, like maybe one is a little behind the other or more, but they may sometimes be correlated. But I think there are times when they're not as well, like cell cycle might not be at all, and there may be other cases. Cell cycle is kind of a special situation because it's so overwhelming as a signal. So I'm not going to deal with it a ton here, but okay. So let me explain a little bit about, or I'm going to try to show you what we do to solve those problems and why I think it's interesting. And I'm showing it on this really nice benchmark. There are very few benchmarks for these kind of data. Mostly we know something about differentiation or something, but there was a nice work done that used metabolic labeling of mRNA to actually get sort of nascent RNA, like very early RNA versus later RNA. You don't, obviously it's a bit discrete, so you don't get continuous time points, but it gives you a sense of what came earlier and what came later, and you can actually kind of get more real values for whether the RNA was produced earlier or later in the cell's life, which helps you get much better models. So we're using this as a kind of benchmark. This was done on hematopoiesis. If you don't know these, so I probably would not know all of these. I don't have my, sorry, I'm just trying to get, I don't have a pointer here for this. That's one problem with doing it this way. Hematopoietic stem cells, different progenitors for the bottoms are differentiated cell types, which I don't expect you to remember. The ones that when we need to know them, I'll point them out. So, but erythrocytes, megakaryocytes, basophils, whatever. Okay, so if you don't use, if you kind of just throw what is probably the best established, most commonly used method, SC-Valo, at this method, it doesn't matter which of the SC-Valo methods you use, they screw up all of them. You will get a picture that basically is generally kind of confusing and mostly wrong, right? So, the megakaryocytes are dedifferentiating, and so are I think that's the basophils are dedifferentiating. Things are just going the wrong directions, right? And so there are probably several reasons why this is happening, but we were sort of thinking, okay, is there a way to fix this at all? Can we do anything with this to get this better? And so one of the things that we did, so I will say that this is totally hacky, right? The right thing to do would be a nice end-to-end model. You infer everything together. We didn't do that at all, partially so that we could actually check that we understood what we were doing at each stage. And there's other reasons as well, but it's definitely imperfect, 100% imperfect, but it still works much better than what we had, which is, it's always good to check against the null. Against the null was pretty good. So, the the first idea is to do some kind of unsupervised learning of these biological processes that we want to infer dynamics for. Like, what are the cells doing? How are the genes grouping together? So have people seen topic modeling before? Okay, so this is like, I brought all of the background for the wrong things. That's what I'm learning for this. Okay, great. So people have seen non-negative matrix factorization before? At least? Okay, so basic idea of non-negative matrix factorization. You have some kind of, say, counts model, right? Some kind of positive matrix, right? Here, you might imagine if we multiply these together, we'd have like cells and genes, right? That's our matrix. And so we have positive counts for the number of transcripts that we identify for each gene in each cell, right? That's like a standard counts matrix for single cell RNA-seq. You might think, okay, I'm going to factorize that into a lower dimension because I don't think that those genes are filling up every inch of gene expression space. They're correlated, they work together, right? And some of it is just noise. So I'm going to try to learn, say, K processes. K could be 20, could be 5, could be, depends how many cells and what you think they're doing. But I'm going to try to associate genes with processes and cells with processes, right? So they both get cast into a lower dimensional space. So then I have essentially a weight for each, here they're called topic, I'll explain that in a minute, a weight for each topic in each cell and a weight for each gene in each topic. And those are, you being of the AI age will understand that this is learned. It's not, but in the old days, right, people would do something like this, but they would use a database of processes where the weights were already known for the genes. And the processes here, we're learning them from the data. Yeah. What's your name?
What's the difference between topic and principal components?
Great question. What was your name?
Guiming Gao.
Guiming.
I also from U Chicago.
Guiming. Yeah, I've seen you. Yeah, Guiming. Nice to see you again. I still don't know your name though, because I'm bad with names. Okay, difference between, so you can think of it, it is similar to a principal component. It isn't orthogonal necessarily, so they're not orthogonal, and they're not maximizing variance. Here, so principal component has like positive and negative values, right? This here, we're just thinking of these as kind of only positive only axes, right? So each gene can only contribute in a positive way and cells can only have positive weights. So that's like the most literal difference. But there's a lot of other ways to think about differences. And one thing is that, there can be multiple right answers, right? And so you could think traditionally, non-negative matrix factorization is really just an optimization. Give me two matrices where I have the least error, for example, if I multiply them together to get one, and I try to recover my original matrix, right? That would be a way of, and there may be multiple choices, but you could try to optimize over that space. Yes, what's your name?
Chimo.
Timo?
Chimo. Complicated one. XIMO.
XIMO. And how I say it?
Chimo.
Chimo.
Yes.
Chimo.
Spanish name from a very specific region of
Chimo, Barcelona.
Valencia.
Valencia, okay. Okay, Chimo, okay.
No, I just have a question. Did you, when you were using this approach, did you ever benchmark it against like cell types that are known to be extremely plastic in their types of gene expression programs like T cells, no? Where even like if you just want to classify them, sometimes it's hard because all of the T cells can do all of the
Are you talking about
programs.
topic modeling?
Yes.
Yes, that is actually how I got obsessed with topic modeling. So I will send you my paper that I first got into it, and there's been other papers obviously since then, but I was working on innate lymphoid cells, which are the cousins of T helper cells that don't have T cell receptors, but they have a lot of the same states. And it was ridiculous because there's like a big cloud of them and they weren't doing any of the things that they were supposed to be doing. And it was super useful to use this kind of sort of decomposition. So yes, I think it's super useful within cluster. It's not that great across cluster all the time because it's not very good at hierarchical decomposition, but it can be very useful when you have a messy cloud and you're trying to understand, that there are sort of differences, there are things going on, but it's not like nice, discrete little worlds, So yes, super useful. Other questions somewhere there? What's your name? Hold on, it's Timo Guiming.
Yeah.
And first guy, it's rough.
Stanley.
Sorry. Okay.
Aditya.
Aditya?
Yeah.
Okay, cool.
Also from U Chicago.
Yeah.
I was wondering, so here what I'm guessing is you have all the cells, like it's all per cell, and then your topic cell X gene matrix is like all the cells, all the genes, right?
Right.
What if you were to, instead of this, let's just say using all cells and then doing topic modeling, would you prefer to do a meta cell, like an aggregate of bin each like group of cells, and then you do SC-Velo or topic modeling? How would that sort of work out?
Okay, so let's come back to the SC-Velo thing. Let's just think about the mixture thing. I actually think we are doing something like that because we are doing this on spatial data right now. And in Visium data, that's exactly what you're doing. You have a metacell essentially that's spatial as opposed to transcriptional, right? So I thought, hey, that's a pretty natural use. It is used, this kind of approach is used for deconvolution of spatial data, for example, sometimes. I don't particularly care about deconvolution because I think you cannot get it right. But just it can be useful as a way of trying to understand how things are correlated and in what ways. I personally really like it in the single cell arena because you're sort of aggregating information across different cells, just like in PCA, but it tends to be much more interpretable. So let me just say the one thing that's different about topic modeling specifically, it comes from an evolution of methods. I'm sorry that I left out all the slides. This is my computer science background, thinking that everybody knows the same stuff I know. But originally there was a kind of an evolution of methods, non-negative matrix factorization. There was this idea that you could factorize the probability that a word appears in a document, right? If you think about these cells as being little bags of mRNA words, right? It's the same thing as like the, websites or journal articles or whatever, right? And you're trying to learn what topics are covered by science or what topics are covered by Wikipedia or something like that, right? And so you could think about trying to learn thematic groups, topics, right? By learning with what probability a word appears in a document, and you'd probably want to get rid of the really common words like and the and whatever, right? And then you would sort of try to learn which topics appear in which documents with what probability, and that might help you understand, the topics then start to have a definition based on the words that appear in them. And the same thing happens with immunology or biology. You're like, oh, I know those genes cause inflammation, so I'm going to call that an inflammatory topic, or a Th1 or Th2 topic or something like that, right? But it's not like it necessarily is the core established definition of that, right? Okay. The other thing is that, so topic modeling basically is a form of this kind of probabilistic factorization where so first of all, you assume that you have like a multinomial distribution that governs the probability of a word appearing in a topic and that topic appearing in a cell. And so you're factorizing that distribution, and the nice thing that makes it super interpretable, but also more difficult to infer than say NMF or to optimize an NMF, is that you're assuming you have, you sum to one, you really have probability distribution both across the words in a topic and across the topics in a cell, right? So it's a two-way sort of probability matrix, and that makes it more constrained and a little bit more difficult to optimize. So some of the methods are better than others, and there's lots of variety in approaches. I will say there's more neural approaches also that we're starting to experiment with, mostly so that we can integrate them into other kinds of larger scale methods. But for actual topic modeling, I found that I can't remember exactly which approach we thought was really good. There's a pretty well established one that's pretty good, but we end up going back to this approach that comes out of Matthew Stevens' lab, who, for people at UChicago, he's a statistician at UChicago, because they did a very nice differential expression. So that's one of the things that is harder to do. If you have a cluster of cells, you could say this gene, is differentially expressed according to a negative binomial gene expression model in this cluster versus that cluster. There is much less work done on trying to understand how to do that for topics where you have probabilities. And so they did some nice work trying to at least establish a framework for differential expression of topics. The reason you might want to do that, right, is because you might not necessarily care about the most highly expressed gene in a topic, just like you don't always care about the most highly expressed gene in a cluster. You care about the things that make it kind of distinct, right? Okay, so this is a whole lot of background I should have included. I'm happy to send more resources. So let me just say that we did, the other thing is just like clustering, there's a topic parameter, K, that I said before, and people always ask, like, how do you choose K? And there's usually many right answers and also some very obviously wrong answers. So I try to weed out the very obviously wrong answers. That's like my method. If you make K very high, you will start seeing lots of subdivision, like the same cells that are sort of getting lots of the same similar topics. The genes start to get split across topics. It just, it looks like you're just sort of slicing the same thing lots of different ways. And so that's usually a sign that it's too high. There are also metrics for this. And then if you do it very low, then you start to see things going together that don't make any sense, that they shouldn't be going together. That does require some domain knowledge. Again, there are metrics that we use. This paper that my student wrote for PNAS, he developed some metrics that also helped us kind of choose this. But anyway, let's say there's really no great way. You just kind of usually something in the general realm of reasonableness will get you there. So we did I think this was 17, I don't remember, 17 topics, something, maybe it was less, I honestly don't remember. But I'm just showing a couple examples. So sometimes topics will look quite distinct like this is a megakaryocyte topic, very high weight at the bottom, right? It looks more or less like a cluster with a little bit of bleeding into the main body, right? So that you'll often get some kind, topics will also, reflect natural clustering. And then if you do sort of this differential expression type analysis, you can look for genes that are released, or even just looking at some of the top weighted genes, you'll see genes that are really associated, and you can check that indeed these are genes that are associated with megakaryocytes, which is good, because if they weren't, we would have not be believing what we're seeing, right? So biology kind of checks out. And then the reason that this becomes important eventually, right, is because say we have this gene that might not actually be the right gene. This might be for a different topic, but anyway, this is a reasonable conceptual idea here, which is that if you to be able to infer these dynamics, you need a reasonable range of unspliced versus spliced counts, right? When you have tons of cells that are not part of a process, right, they have nothing to do with megakaryocytes. They're like way on the other side doing something completely different. You don't get any data from those cells about what the parameters should be for the megakaryocyte genes. But if you focus in on, a gene and the right cells, even if you use, and in fact, you do want to use the cells that are low weight because you want to see the spread, right? But you kind of let it bleed a little, but you focus on the cells that actually have some range, then you get a much nicer range of spliced and unspliced, often, not always, depends on the gene. But you're much more likely to get a good range. Just makes sense. You're concentrating signal relative to noise, and that gives you a much better chance of being able to infer the right parameters for this gene, right? Does that make sense, what I'm saying? So just conceptually, you're getting good inference. Okay, so let me just say, sorry. First step, I didn't show you, but we have lots of other topics. I'm sorry, I had many different versions of this, but some of them are kind of more in the cloud. Anyway, we basically dissect this into a bunch of topic-specific genes. That's the first step. Again, it's a little bit of an art and a little bit of science. Next thing is that we use topic-enriched cells and I call them topic-specific genes, to estimate those velocity parameters for that group, right? So you can imagine we're taking a subset of our big matrix and we're including now our unspliced counts in that matrix, and we're using that to now estimate our RNA velocity parameters. So like you said, we get a different kind of set of parameters for each of those processes, right? And we would have a different model for where the cells are in that process for each one. So here, what we did was super simple. It's just a chemical master equation, which me, even knowing no physics, could understand. And then basically to fit the parameters, we did a simulation and KL divergence with the landscape, and that actually worked pretty well too, and we could do it relatively efficiently. I think that's really a testament to how bad most comp bio methods are, that you can do something like this and it's still better. So not, sorry, that sounds really mean. I just mean the coding is not our strong point, right? So we don't have to be amazing coders, myself included. So basically this is kind of, you know, it's very simple. It uses very simple methods from both the kind of very simple old school machine learning, pretty simple physics for, you know, statistical mechanics kind of ideas, and you put them together and suddenly you get actually something that is much more informative and useful. Yeah.
So going back to the topic modeling, do
Yeah.
you do it on the like normalized counts, or do you do it on the embedded, like PCA embedded?
No, you do it on the counts. There's no, so sorry, the PCA, we never use, so the only time we're using a PCA is basically to create the UMAP that you see, right? That's it. That's the only place I'm using PCAs, to create the neighborhood graph on the UMAP. So the topic decomposition, right? That's at the level of the probabilities. And that's just used to select the cells in the genes. There's lots of debate over how to use, like so officially you should just use the raw counts model. That's what, you know, that's probably what technically makes the most sense. But we did explore using like corrected counts for the length of the cell, for the amount of mRNA in each cell, library corrected. And I'm not right now blanking on which thing we decided to so there are a few small corrections that we might have used. But then we would have had to round to counts, to integers. Yes, somebody, your name.
Hi, my name's Connor.
Connor.
I'm wondering how this all changes if instead of looking at cells within your topic, you use whatever clustering UMAP or whatever embedding you use spits out.
You mean if you use, so most methods use all the cells, or sometimes they will try to go to like at least a subset of cells that they know are developmentally related or something like that, or they'll use like a cluster. And so I can tell you, we did some ablation experiments where we tried to we got rid of the topic modeling, we got rid of the stochastic modeling of the bursts, only because a reviewer made us, and it they both actually help make things better. I think my basic feeling is that if there's a very strong signal of the trajectory in the data, then you don't need stuff like, you don't need to decompose it that much, because the signals, the signal that is dominating everything is the thing that you want to recover. And I think that's part of why a lot of trajectory inference works in developmental systems. I'm going to show you an example if I get to it, about a case where we found it incredibly useful and we couldn't figure out any other way to do it. So, these tend to be a little bit more technical because they're just, they're annoying biological cases that people wouldn't actually know about from the, in the general. What was I going to say? Oh, here, this is just showing that if you don't have the burst model, so this is a comparison with just a one state simple model with including the burst. And the main thing that you can see is that using this burst model allows us to put much more weight at 0, 0, right? It allows us to actually do a better reconstruction, especially of the low end of the probabilities. And I think it's that same sort of reasoning that I was saying before. Okay, so next thing is, this is kind of standard. Some of this at least is taken standard from RNA velocity. Usually there's some goal of reconstructing transition matrix, and from there streamlines and pretty pictures, right? And the transition matrix, usually the way it's reconstructed is this idea, sorry, it's on the bottom down there, but that the probability of a cell going from, say, I to J should be, what is this? No, this is not I to J. What is this? This is going from cell I to cell K, maybe?
Okay, here's the topic.
Oh no, it's going from cell I to cell J. K is the topic. Yeah, sorry. K would be a topic here. Cell I to cell J would be, you could think of K even as being a gene in the original, right? It would be it's the angle between Anyway, it doesn't matter what the notation is. The idea is very simple, right? The idea is I look at the direction I'm going, my velocity, and I look at the vector that's between me from if I'm I and J. And if those are close, then I say my probability is higher to end up near that cell, right? Very simple idea. So if the cosine of the angle is high, then that makes my probability high. So this SJK minus SIK, right, just is the position. That's the difference between the current mature mRNA positions, and the VIK is my velocity vector. So if I look like I'm moving in a direction that matches where another cell is, then I would want to put an arrow in that direction. I will say that this is a little bit dependent on what other cells are there, right? So if I'm at the bleeding edge of something, I'm not going to have great matches for where I'm going. The other thing that happens is that if I'm kind of 6 of one, half a dozen of the other, right, then it becomes hard to even visualize that. And that is where having topics, somebody over there asked me, I think Stanley asked me, topic specific velocities can be really helpful then, because you can actually see more how cells are moving according to one process or another. And when cells are kind of on the fence, they're a mixture, then it's really helpful to be able to see that. Yeah.
Two observations. For topic 3, which I guess is the megakaryocyte identity genes, like you see like a very like almost stepwise process where you are either a megakaryocyte or not, right? Kind of failing in a way to capture the developmental process from whatever megakaryocyte progenitor there is to the megakaryocyte. Topic 7, on the other hand, I don't know what it is, but
this is going towards erythroid cells. Okay.
It's crossing the boundaries between monocytes and
so wait, hold on, let me just say
progenitor and those are very well established developmental boundaries. So unless it's some kind of other type of process.
Yeah, so this is the reason why my student Frank would never let me show these originally, because he was sure that they were going to be super misleading. Then my student Hope did this immune paper with them, and she was like, what is he talking about? These are the most useful things in the world. So there's a caveat here, which we haven't figured out how to actually make apparent, which is that when you look at the streamlines, right, that doesn't tell you about the weight of the topic in the cell. So this topic is probably extremely low weight up here and basically irrelevant to where these cells are going. Does that make sense?
Yeah.
And right now it's sort of showing a little bit, but the range of the topic isn't, you can see the topic weight up there. So maybe it's a little bit relevant. Topic 7, I don't know, but there's also plenty of topics that are up. I will also say that one of the things that happens is that you're looking at a visualization. And so all of the cells that are up here may not actually be fully committed, whatever those cells are, right? As opposed to erythrocytes. What were they? I don't remember. I can go back.
I guess granulocytes and neutrophils were on the left. Yeah, monocytes.
Monocytes, yeah. So they may not be fully committed monocytes, right? There were it's a visualization based on the.
What about the other part, like the fact that by soft clustering these cells, you have isolated maybe such a terminal program,
right?
That somehow by plotting that program, you clearly see that there's no clear continuity with the rest of the cells.
I agree with that. That brings me very nicely to the next thing. So it's true that if you don't capture much of the transition, right, when there's a fast transition or you don't have a lot of cells in a transition, it is really hard to map that transition and you won't have it very well represented. And that's just a fact, right? Like it's very hard to make up for that. The thing is getting those arrows that's actually a pretty strong indicator. So even having a few arrows on the above, like you have to have looked at about 1 million of these plots to know that that's actually a pretty strong signal that there is probability kind of going that direction. But you're right that in general, you don't get that great of a coverage of that transition. So there's a couple of ideas there. The basic idea that we had was like, okay, if I want to generate some general notion of how cells are moving through the space from these various topic velocities, I could just use my topic weights to integrate, in the sense that I take a step according to the probability matrix for each topic, depending on my weight in that topic, right? So you have these different transition matrices, and then, each time you take a step, your step basically is a weighted kind of probability depending on how much weight you have for each topic. You'll take a step in the different transition matrices. So that's the main idea of how we get some kind of global velocity, it works okay, right?
Look at
It's
that.
very dependent on the topic. And it actually works, it gives us a much better picture of that. The other thing is that, so the method is, I think, pretty sensitive to the amount of bleeding you allow. Like how many low weight versus high weight, right? Because in some sense, we're actually doing something pretty stupid, right? We're thresholding on this continuous topic weight to be able to choose the cells, right? Cells can belong to more than one topic. And in fact, you absolutely want them to belong to more than one topic, or else you don't get any cells in that sort of, transition zone, right? Where they're connecting the different trajectories. And if you have all of the cells in all of the, if you don't, then you end up with, you may as well have not done the topic modeling to begin with, right?
Is there a way of like, let's say, for instance, plotting the distribution and see, okay,
Yes.
if topic one
yes.
is clearly by model, then I'm gonna be more lenient and I'm gonna include more of the things that
That's
have a little
exactly
bit of
what he does. He basically does a kind of, you sort of look at what at the different thresholds that happen if you come from above or you come from below, and how it spreads, and you basically try to choose something that's somewhere in the middle. So that's exactly the kind of thing that we do. But of course, again, it's an art, but yeah, you're right. Okay, so basically the point is that without doing, like, we did not do a lot of rejiggering, we did not, do hocus pocus, like we get a pretty good inference without using the metabolic labeling data. So this fixes. So we were psyched because this is actually a pretty tough data set, and it seems to work pretty well. We also explored, we also explored some other ideas that will be familiar to people who have done, any kind of statistical physics, like trying to analyze this transition matrix by looking at the stationary distribution. So we got a better representation of the terminal states in our stationary distribution. We also use mean first passage time, which was this idea that I was really interested in as a way of judging how close cells are that didn't rely on the UMAP. Right? If a cell is near in transition, then you would expect they would have shorter mean first passage times to get from one group to another. It is very finicky. So it works great in this data set, doesn't work great in some of our other data sets where we know some things are true or false. And we don't exactly know why. So I will say, I think it's a great idea. It might be a little sensitive to the sort of noise that we have already, and we haven't had a chance to really investigate that. So I'll just show you this example. Wait, first of all, am I supposed to be done now? Like, no, I still have more time, right?
3:30.
3:30. Okay, great. That clock is so large, it felt like it was. I teach in all these rooms that have no clocks, and so I'm like, it's staring at me angrily. Okay, so for people who are not used to immune jargon, I'm going to try my best to keep it at a minimum, and just give the sort of basic idea of this analysis that we did. So right as we were sort of publishing this topic VLO work, my lab was collaborating with a lab of a close colleague, and he does developmental immunology, which is not a part of immunology I particularly ever got into. But the question there was about understanding, he's interested in understanding the effects of different immune components. And for mouse models, it's super helpful to be able to delete them, right? And to delete them, it's super helpful to have progenitor, at least transcription factors, right? That mark certain progenitor populations so that that this population leads to these cell types so that you can actually manipulate better. So he was looking for understanding the difference between NK cells, which hopefully some of you have heard of, natural killer cells, very important in fighting cancer and lots of other immunity. And ILC1s, which probably very few of you have heard of. It's a kind of like a innate version of an NK cell that does a lot of the same things that NK cell does sometimes, and sometimes are very different. And honestly, they can be very hard to tell apart. The main reason that we know they're different is because they are connected. So these ILC1s are connected to a bunch of other kinds of cells that, are very different. And so we've understood a little bit about their development and how they arise, but it's pretty hard to actually understand how these arise differently. And so they were basically trying to understand this. So you can see here, I'm just focusing on a smaller part of a larger UMAP where we're taking all these mouse progenitor cells, ILC1s, like all collected from one bone marrow experiment. And I've highlighted here the NK cells and the ILC1s. And you can see that these are pretty different. They have a few things that mark them, but they're not that easy to tell the difference otherwise. And so first we sort of noticed that there was, okay. So first of all, let me say, I haven't shown you, but if you do a sort of similarity-based analysis of this, right, which is pretty much the only thing you can do because we don't have any staged, like you cannot get a longitudinal version of this. We can't knock it out. These are all present, right? So if you try to do some kind of a similarity-based trajectory analysis, it's like, it looks almost like a complete graph or something, right? Like things are all very close. There's very thick edges between things that that didn't arise, one arose from the other, but you don't know which way. And in some cases, neither of them arose from each other. They arose from something above them, but they're so similar that, that kind of method is not going to be helpful for telling you apart. So you're kind of looking in a very fine-grained way at very similar cell types. And for there, it's really nice to have direction, right? Like to know how things are changing. Yes.
No, sure, but it harkens back to the question I asked at the very beginning. No?
Yeah.
I was even talking about worse T cell states, like when you just have a bunch of CD8s and CD4s, and they don't even have defined TH states, and they have like more interferon response, effector response.
This isn't the example I was
similar to this.
Yeah.
I always wonder, like, wouldn't it be like to take some of these programs are response programs? No, they're like there's some molecule in the environment, and then they trigger that response. And some of them might be related to some developmental process or something, some defined set of state transitions. Would it be any good to just like using your approach, essentially weeding out which topics belong to the category of responses and processes, and which topics belong, and then just focus on those which have developmental or process signal versus those that are clear pathways.
Right. So first of all, sorry, I wasn't very clear, but this isn't the paper I was thinking of, because this is absolutely not functional response. I was thinking of a different paper where we did a lot of ILC work that was functional response. This is very developmental. And I will say here, one of the things that's hard is that some of these cells have what we would think of as a functional response, but at baseline, right? That's like how they, and so it's possible. We didn't try this. It's possible. I think it can be hard to distinguish those kind of things. So, you could say that something is functional, but actually could be important in terms of the developmental trajectory as well. We know of examples like that, right? So, but anyway, we can come back and talk. I don't want to lose everybody here on the immunology, but I would love to talk more. Maybe I'm also not fully understanding. Anyway, the point was that we did this sort of velocity analysis of these cells. So here we didn't focus in just on one topic. We focused on any topics that were sort of closely related to any of these specific sort of precursors, ILC1s, NKs. Like we left out, there were some other progenitors, other ones that were very distinct. We left those out. So this isn't just one topic, but it's also not like all the topics that were in the data. And one thing that we noticed is that it looked like there was this sort of group of cells that, like I said, they're not going directly into the NK cells, but I will say that my experience with UMAPs is that they really like to cluster. So often, if you did a diffusion map of this, you probably see a nice, long, continuous path from one to the other, because diffusion maps really like to spread things out along transitions. And sometimes things look very distinct in a UMAP that aren't that distinct. So I sort of was like, there's this group of cells that look kind of like they're feeding into that. And they were positive for a specific progenitor or a specific transcription factor, EOMES. And they already had this sort of theory that EOMES might be important. And so this was already kind of a progenitor. But what was even, so here, this was already something that was kind of a little bit in progress. And so this was helpful because we kind of could confirm that we were not doing trash, right? So they already were in process doing bone marrow transfer experiments, and they confirmed that these EOMES positive progenitors are pretty committed to the NK lineage. Like maybe not 100%, but 90%, say, or 95%, right, of them become NK. However, there's this other group that was like a little bit above, and this was interesting because this has this other very important transcription factor, PLZF. And it turns out that cells that are positive for both of these are actually dual progenitors. I realize that none of us necessarily care about who's going to become what cell type in this specific immune system. But the point is that it gives you a handle on how to control downstream cell fate, transitions. If you know that, that to be able to develop into an ILC1 or an NK cell, these are the key progenitors that impact these. And using those same kind of bone marrow transfer experiments, which will make you sad if you don't talk about mouse experiments too much, that we can confirm that actually, like, it's not 50-50, it's like 40-60 or something, but a good percentage of those cells go one way and the other go the other way. So you recover a really good distribution of the cells. Okay, so this was just one example, more recent. I think part of why this was interesting was because it was something where, like, we didn't know directionality, right? And if you just look at this in terms of similarity, it's just totally unhelpful. Like, you don't know where things are coming. And the transitions, you have all of the cells represented. You can't control the transitions very well. And so I think there are, I think that this is representative of the kind of situations in which velocity is important, but maybe not limited to only this kind of situation. This is still developmental, let's say, and I think there are plenty of other kinds of examples. Sorry, there's the PLZF. So just for a minute, I want to talk about, I think, at least, my concept of why I think spatial would be interesting. Like I said, right, we can think of spatial data. So this is the kind of data where you might have mRNA for each single cell, or mRNA for spots, which tend to be somewhere in the range of 1 to 10 cells at a time. There's spatial layout. And you get somewhere either between like 500 or genes or like the full transcriptome. To me, this seems like a really natural place to actually care about instantaneous change. Like, we care about early changes in cells. We care about knowing, when does an immune cell that's been cytotoxic stop being so cytotoxic? We also care, of course, if they don't go into the tumor environment. Like, there's lots of other reasons why cells could be less effective in a tumor environment, but what's the difference between an inflammatory process that resolves in a gut epithelium and one that becomes like, IBD and doesn't resolve, right? Are there ways of understanding, even though the epithelium is turning over and over and over, are there ways of understanding how the immune cells are getting programmed by their environment to sort of respond in a certain way? It's obviously a very big and complex question, but it strikes me as like a really good use case for this kind of instantaneous change. So that's something I've been thinking a lot about and working on. The idea would be, can we associate cell-cell neighborhood, our interactions with dynamics? How do we see different kind of trajectories change in different environments? And there is some existing work in this area. We have messed around with it and found it, I would say, not definitive, would be the nice version of that. Again, this is all very buggy, so it's really hard to do work that generalizes everywhere, but I think it's a hard problem and it isn't solved yet. Another thing that would be really cool to do eventually, right, is to have other kinds of contexts, like metabolites, other kinds of signaling molecules, proteins, even cellular features that you can get by imaging. Like, it would be a nice way to be able to use some of this multimodal data that is getting gathered in a spatial context now. One of the things that we've noticed, though, is that some of these methods, right, so why can't you just do this directly in spatial? Like, why not just apply this directly to the spatial data? Almost all of the more commonly used spatial methods don't get any of the unspliced transcripts because they are using either probes to try to tag specific transcripts or they weed it out on purpose, or the tissue gets processed in such a way that they don't survive, or I'm not exactly sure, but many of them do not get it. Even ones where you might have seen a paper where they claim that it did, we can't find any reasonable signal for it. So what that suggests to me is that I mean, either you can restrict only to the few methods where they actually gather that kind of data, right? And we can test there. But a more interesting idea, right, would be to do some kind of inference about that uses the information that we can glean from about velocity from single cell with the information that we can gather about neighborhood from the spatial, right? That's a pretty classic idea. Of course, I've spent all this time telling you that you should think about which cells you're going to learn parameters from, and that might be true in terms of the spatial too, right? So it may be that you don't want to learn from all the monocytes, you only want to learn from some of the monocytes that are in a spatial. What we've seen so far is that actually when you do straight up topic modeling in spatial data, you actually get pretty good recovery of a lot of spatial signal. And so we're hopeful that that's, that you can actually sort of put together in some kind of integrated way, a sort of topic model from spatial and a sort of topic model from single cell, and use that to do some kind of joint inference. There are lots of ways that one could do this, and people have tried. So that is really the open question that I think is difficult, but I can show you maybe an early example if you want, or we could stop here, whatever you guys want to do. Do you want to see an example of this? Oh, yeah. Yes, no.
Yes.
Okay, all right, fine. So I'll show you, I'm going to skip this part where I do the acknowledgements, and I'll come back to that. So this is I'm going to probably talk about this, and I talk about, you'll see it twice, and then you'll understand it the second time. It always takes me 5 times. So, okay, but this is just to tell you a little bit about the data, right? We've got, we've tried this in existing data as well, but this is the data we understand the best because it was gathered by my student with Tom Gajewski, who's a cancer immunologist at AU Chicago. So they're using, this is this very simple colorectal cancer model in mice. And here we've got H&E histology just showing that what the tumor kind of vaguely looks like. And you have this tumor core where everything's kind of necrotic. And we've also collected, so we have single cell and spatial data, both. This is the single cell data. So right here, I'm going to focus on the treated cells. She's studying immunotherapy and other kinds of techniques for controlling the immune response to the tumors. So here, this is very simple, PD-L1. Many of you have probably already heard of that, right? It's a kind of, it sort of helps the immune system fight the cancer. And so when these data actually look much more consistent than without treatment because they're sort of forcing the system in the same direction. So we started with the treatment. It's actually a little bit easier to deal with. Okay, so let me just give you, if you haven't seen spatial data before, this is using an older method that has not been maintained. It's a great method, but hasn't been maintained. So, this is kind of old data, but it gives you a sense of what you can do. So here's what's called a spatial factor. You can think of it as sort of like a non-topic or non-negative matrix factor that is, but where, there's a bit of a prior, there's more emphasis on contiguous cells or spots having the same topic. So you try to infer topics that have some sort of spatial continuity, or at least those have a higher score, say. And so here's an example of a factor that we think captures tumor-associated macrophages based on the cells that are upweighted in it. And if you actually plot the factor, you can see it kind of lining this very specific region. And this is in crappy old Visium data. So it's 10 cells at a time. It's not super precise. It's kind of amazing that you can get this kind of resolution. And basically we said, okay, let's use those genes. We can plot that same we can make a gene score, even though we don't have a joint embedding just for rudimentary purposes. We can try to relate the single cell and the spatial by creating a gene score using the genes that are in the spatial factor and just coloring the single cells by that gene score. And if you do that, you can see that it's very specific to just the macrophages. Sorry, I don't have a pointer again, but it's just coloring the macrophages, not any of the other cell types. So we felt a little bit more confident about that. I wanted to say that we also learned from doing this that it seems really important to also let spatial information influence influence the model, although we haven't figured out exactly how to do this. But many people think of single cells like the Bible, and spatial is an instance of something, but there are things that are hard to get out of single cell because of the dissociation. So this is an example of a spatial factor that has, again, a pretty clear sort of spatial location a little bit inwards. It really looks like it's capturing cancer-associated fibroblasts, and we have like one cell that has a tiny bit of weight for the cancer-associated fibroblasts because they just get removed by the treatment. Yes.
That's weird. That's weird. Like usually they
What, the fibroblasts getting removed?
Yeah.
It depends on the preparation. It's pretty common for them to be. They're very sticky.
What,
so-
what model is it? Is MC38? What?
Yeah, it's MC30.
Because it responds too good for it.
Yeah,
It
exactly.
looks too good for the
Exactly.
response.
Please do not harass me about the model.
Okay, okay.
We know it's not a perfect model. It's just a.
No, I'm just curious because
Yeah.
sometimes like some tumor states are fibroblast-like and they can be confounded with true fibroblasts. So not sure about MC38. Haven't looked at it much.
I mean, it's
but-
possible. What I will say, what's important is only this, that we have the non-immune component here, right? What's important is just that those genes, the cells expressing those genes don't show up in the single cell data. So they get removed during dissociation, right? Maybe they die more easily. Maybe they're stuck to other stuff. Could be lots of technical reasons. It's just all I'm saying is that they're clearly there
Do you
and they're
think
not
it's
getting
related to the
captured.
fact that in your spatial data, your H&E the first slide, it shows this, it's lining this way because it shows a region that is probably not tumor, right? And then the tumor is surrounding that area. Do you think that it could be related to that, to how you collected the tumor versus this particular slide?
It's possible. We do see it in other areas too. And I would say that, the fibroblast kind of being tumor adjacent is not unheard of.
Yeah.
We didn't delve a lot into this. It just made us think, okay, there are signals that come through in the we know that, right? That different technologies tend to show better certain kinds of data than others. So that's mainly what I'm taking from that. Not so much, I think there are ways to change the preparation so that you don't lose fibroblasts and we have,
Maybe just
The other thing
the
is-
tumor that you're cutting is the part of the tumor that you're cutting. Like I've never
She
had
did. So we definitely did this with several. It's not like we only have one sample,
Interesting.
right? So
It's like very solid tumors, and you're not really getting the stroma, and the stroma is here always surrounding the tumor, quite not infiltrating.
I'll have to ask my student who collected it. We haven't written that part yet. Sorry. I know they can be kind of squishy, but I think the treated ones are usually okay. They're small, but the big ones are very squishy, I think. Sorry, I just want to show you a little bit because I don't want to lose. What was your question?
Very quickly, sorry.
Yeah.
I was
No, that's okay. What was your name?
I'm Sui. I'm
Sui, yeah.
one of the speakers here, but I was curious about this.
Sure.
Fantastic.
Wonderful.
No, it looked like your macrophages and fibroblasts are co-localized, right? Is that?
Yeah, it does. I mean, they're not exactly in the same area. This thing is, they're like a little bit inward, right? So, I mean, what we think is the tumor core. There's probably necrotic tissue in there that could, I'm not over interpreting that. But what there is definitely spatial kind of localization, right, of certain signals. Let's say that from a mathematical point of view, you can just say like, okay, they're topics, they have spatial regionalization, say that much, right? But I agree that what it means would need a lot more investigation. You can also go the other way, right? This is just to show, okay, if we did a topic model in just the tumor-associated macrophages, just those top cells, we did like a 3-topic model in them. And then we colored the cell. We did the same thing in reverse, right? We made a gene score and we colored the spots in spatial with those. And you can see that these different, what we think are tumor-associated macrophages, have slightly different localization. And do we know exactly that this is real? Absolutely not. None of this has been vetted. This is all like, very preliminary. But it suggests that, it suggests that there are some differences, at least in terms of how local these different groups of cells are potentially. Okay, so let me just say what we wanted to do, because what we did is a hack, but I think the idea of it, the results suggest that you could do something more interesting. Okay, so what I said to myself is like, okay, what I really want to know is similar to this question with the students, right? Changing if they, if I put an experimental student with a computational student, they start talking to each other and they start moving each other, right? I want to know if the cells that are in a certain area, if they're actually changing more, or if they're changing in some direction towards other cells that are spatially oriented, or are they just changing every which way, right? Like, or are they not changing at all? Right? If you do this, there are plenty of cells that are not changing particularly, right? They're not strongly upregulating or downregulating anything, but you could ask that question, right? That's what I think would be the interesting thing to ask. And we've done this sort of a little bit more. And I think it's interesting. I mean, I think we happen to focus on this, but I think what's interesting is that in this model, the macrophages are some of the most interesting cells to focus on because they're the most dynamic. So basically we looked at, you can kind of see here, right? Oh, sorry, I'm going to come on this side. This is a grid-like model instead of the streamline because it's a little more accurate. But basically you could see here that we inferred this velocity through this macrophage this is the UMAP just isolated to the macrophages. And this is the colors of the sort of cells are smoothed over topic 3 weights. And the arrows reflect the overall sort of inference of the RNA velocity transition matrix for the macrophages altogether. And you can see that many of them don't look like they're going anywhere. And then there's this sort of group that looks like it's like pretty strongly. Okay, so regardless of whether you believe that, let's suppose that we did something right and it's right. The question I had is like, are these organized in some way spatially? Are the ones that are at one end different from the ones that are at another end? And so the thing that I said, okay, if I can do nothing else, I have like a few days, I want to understand this. I can make a gene score that basically I divided up the cells that are here, here, and here. And I made a differential expression gene score for each of those cells. And then I checked that it wasn't expressed generally, right? Like I checked that I could narrow it down to genes that were not expressed by the other cells because I didn't want to get confused. And fortunately, I could do that. You very well might have cases where you can't do that. So, and then I just said, okay, let me just color. So you can see I checked that I could do this, right? So I colored these, they're colored orange if they're high in the early gene score. I colored them dark if they're colored late, and the pink is sort of somewhere if they're both. I think it was if they're high in both, right? So they tend to fall in the between. I just split it into 2 actually, and then I had this group in the middle. And then I just said, okay, is there some spatial organization? You know, if you squint, it looks like, right, the orange ones are kind of more towards the tumor, and the darker blue ones are sort of more towards the core, right? So if you squint, you might sort of guess that like there's a movement. I don't think the cells are necessarily moving, but there's a movement so that the transcriptional profiles of the macrophages are changing so that the ones that are sort of more close to the tumor are becoming more like the ones that are kind of in the core, right? And so that would suggest that there's an evolution that way, which would be not crazy, right? And the fact that you could detect that is nice, right? So it's a kind of a proof of concept. It doesn't mean it's right, doesn't mean those genes are right, but it's like a proof of concept that one could actually infer this kind of signal, and hopefully you could do it better if you were able to pull information across different slides, across different, right? So that's one thing that topic modeling does not have a lot of work on is, for example, controlling for batch, including information across different experiments and different slides. You know, there's all kind of like weird infrastructure that we take for granted and other kinds of statistics that isn't as much there. So just thinking about how to use all of that. Does that mean my time is up? So like just in terms of if the spatial, I think we went over this before. So this is I wasn't sure if I was going to get to this. So, right. So I think this is an interesting area. I think there's a lot of work to be done here, both on this sort of framework and the AI side. And we'd like to be able to, you know, potentially leverage foundation models, although it's debatable to me when they're sort of the most useful. Hold on, I'm just going to come back 1 second. I just want to make sure that I close out here. Okay, so recap. I think most important is like focusing on the right places, the right cells and genes to learn signal can be really helpful for improving inference. You can take relatively simple methods. You know, there's been some recent work showing that a lot of the biggest foundation models for single cell like don't do better than PCA and linear regression and things like that. So hopefully at some point that may not continue to be true, but essentially simple models capture a lot and they're much more interpretable, and that's why we stuck with them so far. I think it's a good starting point, but it is also harder. It becomes harder to do, you know, use a lot of data and use them in sort of more complex ways. So we're now moving out from that. And there's a ton of prior work in all of this, in matrix factorization, topic modeling, their use in biology or in other areas, dynamics, There's been a ton of work in RNA velocity. There's was an explosion for a little while, including using information like that would get at more of the gene regulation. Like you shouldn't be able to, assume that you're getting upregulated if the thing you know that you need to be upregulated isn't expressed by any other cell, right? So just kind of using some basic regulatory logic and protein or chromatin accessibility data and so on. I didn't cover any of that. So there's a lot that I missed here. And then this feature that I think is interesting with spatial. And just my acknowledgements. So I'm using this particular picture because it shows Frank, who's now at a biotech venture capital. So if you have a company idea, you should talk to him. But he's in New York. And Hope, who is she's in process of graduating this summer and looking for postdocs. And Rixandra is the one who she's still a grad student with me, and Tom Gajewski. Suri is in the chemistry department at UChicago and was Frank's co-advisor. Bana and Ben McDonald worked with me on the paper about the NK cells and the ILCs. They're immunology collaborators, and Bana was Hope's co-advisor. I think that pretty much, and Zitao was the lead author. He collected all the data for the ILCs paper as well, but he's graduated. And all the people in red are gone now. So, okay, I think that's everything. Any last questions in our final minute here? Yes, what's your name?
Lila.
Lila, okay, great.
Just really quick, I think I might have missed it, but you mentioned part of what you were doing was you were trying to account for like genic bursts.
Yeah,
can you just
and I didn't talk about that at all, yes.
Can you, just like really fast explain how you did that?
Yeah, so we basically changed the model so that we were using a geometric distribution of bursts. Basically, the size of the burst was geometrically distributed, and we inferred the parameter for that along with the other parameters. So it added another dimension in terms of the landscape that we were fitting. That's not a very complete answer, but basically. Then when you sort of when you simulate, you also simulate bursts according to that burst parameter. So instead of simulating, if for a given rate, you would simulate bursts that essentially have an average of a certain kind of transcription rate. That's sort of, you can think of them as being kind of equivalent to a constant rate model, except it's on average instead. Any other, and thank you, I'm sorry, I totally dropped that.
So in
what's your name?
Avinash.
Avinash.
Yes.
Nice.
In your spatial data, so maybe I missed this. In your you use the topic modeling for the inference of that. Do you need RNA velocity information from like the spliced unspliced count, or do you not need that for
the topic modeling of the spatial?
Yeah.
Well, we would love to have it, but for the most part, we don't have it.
Okay.
So what we're working on right now is some kind of joint embedding.
Okay. And then inferring it from the single cell.
Yeah, you could do things lots of different ways. I've seen some people, like, you could try to learn everything in the single cell and project it onto the spatial. You could try to learn a joint embedding and figure out. We're experimenting with existing methods and also finding them insufficient, trying to figure out what would be better, which is, the easy part is the criticism, the hard part is the bettering. Somebody else had a question. What's your name?
Nice to meet you, Mahsa.
Masa?
Yeah.
Nice to meet you. Yeah.
I have a question. I think I didn't get it. How we could understand that this type of cells will not change randomly in another direction or trajectory? How we can understand?
You mean like just kind of noise sort of thing? Yeah.
And how
totally could. I think they totally could. I think the idea here is that the inference, so the way that we're sort of thinking about it is that you may, like, suppose I have a few topics that tend to be shared, at least they have some overlap. So I might try to learn dynamics among cells with those topics, right? And if they are mostly static or kind of random, I don't learn too much in terms of a coherent set of dynamics. When there's a sort of coherent change from one to another, that's when I tend to see kind of consistent upregulation of like if they're moving from this into this phenotype, then you see consistent increase of transcription of the genes that are here and consistent decrease of the genes that are here, right? And you sort of see them going that way. So it really does require sufficient numbers of cells doing the same thing to be able to get that signal. And if you don't, then you'll see noise, and it's possible that you would infer noise to be signal when it's not. So that is very much a possibility. We do try to look a little bit at the genes and confirm that at least we're recovering some of the things that are known to happen and not recovering too much that's too crazy. But it's that dance of like partial knowledge, right? And I think it's where I really am very glad to work with experimentalists who can validate things, because otherwise I wouldn't believe anything, essentially, right? It makes nice pictures. But anyway, yeah. What's your name?
Hi, I'm Tarak.
Tarak.
Yeah. So I have two questions regarding the spatial data. One is like, so the niches or the domains that you found from those two factors that you mentioned.
Yeah.
Like, so the thing is that, is there a way to complement those findings using something like ligand receptor interaction so that you really quantify if they're communicating with each other?
Yeah.
And the second question is, do you want the second question now or?
Go ahead. If I forget, you can remind me.
Yeah. And the second question is about the joint embedding that you are trying to mention. So I just like to understand like how you are planning to do that, because there are like you need to kind of account for the spatial closedness plus the closedness in the transcriptional space. So is that do you want to like use a 2-level like graph neural network, something like that? I'm not sure.
Great question. So I have a whole part of my lab that's been exploring just spatial methods for analysis, spatial topical kind of ideas. I don't know if you want to consider it reassuring or not reassuring, but basically a lot of our initial experiments suggest that you can do quite a lot with non-spatial methods. And so that would suggest that potentially you could actually do a joint embedding. They will look different because they definitely have different traits in terms of like which genes are detected and how much of the genes are detected. But you might be able to ignore the actual spatial neighborhoods and still retain a lot of the spatial signal potentially. So, but most of our initial ideas were all about retaining and doing like you said, it's sort of a dual kind of
I think
loss.
that probably what you are doing might be tied to the question that you are trying to answer.
Yeah.
Let's say I walk a little bit in the glioblastoma space, and there it's extremely important for you to understand the immune microenvironment.
Yeah,
and
we thought a lot about glioblastoma. Actually.
Yeah. So in that case, probably like keeping the spatial data intact and accounting for them.
By the way, I don't mean to say that the spatial data isn't important. What I mean to say is that it is already imprinted so much in the transcriptional profiles that you don't lose it when you physically don't keep the neighborhood relationships, the actual spatial information. But I agree, it's super important. I think that's really an interesting phenomenon. And we could talk more about it. I presumably that would not always be true. It may just be true because the information is pretty coarse right now or something like that. Right? It may depend. The first question you asked?
It was about the niches and correlating them and complementing them with receptor.
Great idea. We agree. Although what I would say is that I don't know that they're necessarily communicating with each other. They could be they could just be seeing similar kind of influences that are in the environment. They could just be at different sort of stages in there, or they could be responding to other signals. Like I don't know that it's necessarily because of them interacting with each other. But like we are definitely interested in sort of ligand receptor type analysis where there's lots of, I think any analysis that brings in anything confirmable would also be really useful. So yes, agree with that.
I think that LR interaction those studies shouldn't be just limited to the individual niches, rather like inter-niche communication.
100%.
I think there probably the LR interactions become somewhat more important to understand.
I'd be happy to talk more about it. Yeah. Okay, does anyone besides Chimo have a question? Okay, you can close this out.
I just was curious.
Now you see how I teach.
On the topics, the macrophages.
Yeah.
One of the topics you showed was a monocyte topic.
Yeah.
Was that the early topic that you defined?
Possible. It's possible. I think there is a monocyte maturation kind of process that we recovered in there. So I agree with that. I think there is a monocyte. It wasn't tumor-associated macrophages was loose because I think there are.
It's continuity.
Yeah, I think there was tumor. Yes, that was what I also thought. I will say that I also think that data is a little bit wrong. So don't believe it too much because you know, it was generated very quickly for a grant proposal. I think there's probably something. I think this is being recorded for posterity. It wasn't intentionally wrong. I'm just saying it hasn't been thoroughly vetted. It's not like published or something. So, you know, don't put too much store by it. It needs a lot more iterations. Say, yeah.
My question is exactly on that. It looks like you term 0 and term 1. Term 0 looks like M1 macrophage and term 1 look like M2. Correct or
We
no?
would love to see M1, M2. We did
I've
not
never
see.
seen that.
Yeah, I mean, I will say I'm also skeptical because you don't see clear M1, M2 that often. But we this is a, you know, this is of interest to my student for sure. We're aware of them. And we originally, all of my other student slides were like, they're M1 going to M2. I think it's monocyte maturation. I actually think it's more of that. Whatever fake signal we might have recovered, I think it's more about monocyte maturation. But I don't think that the velocity is fake. I just think that the way we processed everything, it just all needs to be vetted and reconsidered. I think those are actually the cells that are moving the most. They clearly have the most unspliced. Like there is a lot of dynamic there, but I wouldn't stake my, you know, job on like exactly what they're doing because that I feel less sure about right now. So yeah, it was just to, I think the illustration of the idea is right. The exact details are probably not right. So don't get too hung up on this. Okay, I feel like I should end mostly because I have an Uber that's coming at some point. But it's so fun to talk with all of you. I can remember like 20% of your names, but at least I have some of them in there somewhere. And I really, really appreciate you making this more fun for me by interacting with me. So thanks a lot. Are you guys coming to the workshop this week? Some of you?
Yeah.
Okay, wonderful. Great. It'll be fun. Okay. Bye.